So the plan is today we're gonna finish the notes. Monday review, test on Tuesday. Monday you will get back. I'm gonna grade your pattern matching activity over the weekend, so you'll get that back, and we'll go over that and talk about that as part of our review on Monday. Okay, so you will have that back. Remember your review sheet is due the day you come in to take the test, which is Tuesday, so you need to have that done. So Monday, for the review sheet, you need to come prepared for a review. So I find that oftentimes students, when they are reviewing for a test in class, I find that students take a more passive role where they want the teacher to just regurgitate all the information, and it's the teacher's role to do that. My take on it is it's review for you. So you need to prepare kind of for a review and help me help you by asking questions and knowing what you want to go over and so on. For this class, the amount of material for a review sheet, I can't reteach everything right before the test, all right? So we need to narrow it down. So that's what I'm gonna ask you to do, is come with specific questions, things that you want to review or weren't sure about. That means that you have to prepare for it, okay? So that's what I'm asking you to do for Monday, all right? So let's finish up this chapter here. So this is where we left off yesterday. We had wrote about tertiary structure. Remember, we were talking about primary, secondary, and tertiary structure. So primary was just the chain of amino acids. Secondary was the alpha helix and the beta pleated sheets that, remember, are stabilized by hydrogen bonds. And then tertiary is when you get folds. So whenever you see, like in this protein here, this is the primary structure is actually in this because you have the chain of amino acids. You also have secondary structure with the alpha helix and the beta pleated sheets, but then you also have it curving on one another. It's not like straight like this, like a helix and then a pleated sheet and so on. You have it curving, and that curving and giving it that three-dimensional shape is stabilized by interactions between R groups. So you could have an amino acid here and an amino acid here, and the R groups interacting to cause this curve to happen and therefore help with the shape. And the types of interactions are these four here that's on this picture. So the first one is hydrogen bond. So what this purple line is a chain of amino acids. So right here there's an amino acid, and this is the R group for that amino acid. So right here, that's the R group for that particular amino acid. And then this amino acid is hooked to another, hooked to another, hooked to another, and this is another amino acid, and this is its R group right here. And then a chain of amino acids here, the amino acid here, this is its R group, and you gotta get the picture here. But notice here, this amino acid's R group interacts all the way down, so there's this whole chain of amino acids, and eventually you get an amino acid here that this is its R group right here. And this amino acid and this amino acid's R group are interacting and forming a hydrogen bond with one another. So that's your dot, dot, dot there. So that helps to stabilize this curve. If this amino acid in this spot had a different R group that didn't hydrogen bond with the R group of this, then you may not have this curve here. All right, so it depends on the R groups and whether or not they, in this case here, make a hydrogen bond, whether or not this curve is going to happen. So then when we look at this second type, this hydrophobic interactions here, this is the second type of R group interaction. Notice here, the R group of this amino acid and this amino acid, they're both what? Hydrocarbons. What do we know about hydrocarbons? They're nonpolar, right? So these guys here, the R groups, are nonpolar. Now let's think about this. This proteins in living systems are in a water-based solution, right? Like inside or outside the cell, it's a water-based solution. So let's say we have water, all right, just water molecules out here. Is water gonna want to have anything to do with those R groups? No, it's not attracted at all because they're nonpolar, which means that they don't have a charge. So therefore, when you put a protein into water, the water will be attracted to the polar parts of the molecule, but not to the nonpolar parts. So what happens is amino acids that have R groups that are hydrophobic, oftentimes that chain of amino acids, the hydrophobic R groups orient themselves in the interior of the molecule away from the water. So these guys are found in the interior of the protein. And why? Because they're hydrophobic. They're nonpolar. So these guys aren't bonded to one another. They just orient themselves away from the water. A third type of interaction between the R groups, this is the third one, the disulfide bridges. So here's an amino acid here in its R group, and the amino acid here in this R group. This right here, this bond between them, I'm going to highlight this bond. That bond, this is a covalent bond. This means that these two sulfur atoms are actually sharing electrons. They're actually bonded together, those atoms. This is very strong. This is a much stronger bond between the atoms than the hydrogen bond over here. Remember, hydrogen bonds are just the attraction of two different molecules due to opposite charges. These guys are actually sharing electrons, and those atoms are physically bonded together. So this makes this very strong. So proteins that have a lot of these disulfide bridges are really, really tough proteins. So the horns of animals, all right, I have that in there. They're very, very tough. They have a lot of those covalent bonds. It's hard to break it down. And then the last one, the ionic bond here, is these guys are attracted to one another because of the opposite charges. And so here's an amino acid here, and this is its R group. Notice here, it has a positive charge. What group is this? Amino group, all right? That has already acted as a base by accepting the H pluses. And here's an amino acid with its R group right here. This has the carboxyl group that has lost its H plus. This has a negative charge, and these guys are attracted because of the opposite charges, all right? And so therefore, this second curve here is stabilized by these two groups of amino acids, there are groups bonding. And so, can you see that if I switched this amino acid with another amino acid with a different R group, they may not bond, and therefore you might lose the shape. All right, so the R group bonding is very important ultimately in the three-dimensional shape of the protein. And the shape of the protein determines the what of the protein? The function of the protein. All right, and so that's the tertiary structure. Now some proteins are done here, and they're done with the tertiary structure, some are not. What do I do with my clicker here? Lost my clicker. All right. So let's look at the quaternary structure. Some proteins go into this. This is the fourth. Whoops. This is, remember, the multiple polypeptide chains. And so an example, I haven't talked about collagen yet. Yesterday I used the example of hemoglobin. And collagen is another type of protein that exhibits more than one polypeptide chain. And then hemoglobin, just a review from yesterday, has the 4 polypeptide chains. 2 of them are the same, so two of them are called alpha, they're the same polypeptide, same type of polypeptide chain, and 2 of them are called beta. And so when we look at this is showing you just a visual. Here's the collagen with the 3 polypeptide chains. Notice here the 3 polypeptide chains weave, and so that's why they call it fibrous. It kind of looks like a fiber. And the hemoglobin, as I talked about yesterday, has multiple subunits. In this case here, there's 4. There's 1, 2, those are the beta chains, and 3, 4. And all those chains coming together make your full hemoglobin molecule. So you have to have 4 of those. So let me just okay, so let me show you how ultimately the shape of the protein really is determined by that first structure, the primary structure. I'm going to show you that with an example of what happens when there's a change in the chain of amino acids, and it can, one amino acid chain in one protein can cause a full disease, which is, I'm going to use the example of sickle cell disease, or sometimes called sickle cell anemia. And so we can have a slight change in that chain of amino acids, that primary structure, can affect the protein's conformation and ability to function. Conformation means shape, right? So shape determines the function. And sickle cell disease is inherited, so we get it from genetics through our parents. It's a blood disorder, you'll see that in just a second, that results from a single amino acid substitution in hemoglobin. What is that? Well, a single amino acid substitution. So you have a chain of amino acids. That means one amino acid is substituted for another. So you just switch one out for the other. And that one switch causes a whole disease that can be life-threatening. So let's look at why that is. So this one change causes abnormal hemoglobin molecules. These two pictures are red blood cells. Remember, hemoglobin is found in your red blood cells, and it's important for carrying oxygen from your lungs to the rest of your cells of your body. This is a normal red blood cell and with normal hemoglobin and can carry oxygen. Individuals that have sickle cell disease and have that substitution, their hemoglobin molecules are a different shape, and therefore their red blood cells are a different shape, they're sickle shaped, and this interferes with its ability to carry oxygen. And so therefore oxygen is needed for your cells because it is needed for you to break down glucose to get energy, and so therefore it's a big deal for the cells not to get oxygen and therefore not be able to break down glucose to get energy. So this relates it to the primary, secondary, tertiary, and quaternary structure. So right here, this is normal hemoglobin and sickle cell hemoglobin. So let's look at this. Here's the primary structure. So this is the first 3 letters of the amino acid sequence. Remember, does the alpha helix and beta pleated sheets and folds on each other to get your 3-dimensional tertiary structure. Then hemoglobin is 4 subunits, right? So this disease affects the making of what's called the beta subunit. So this is normal, so it has the normal shape, and then the 2 alpha polypeptides come together, and you have your normal hemoglobin. Notice it says molecules do not associate with one another, and each carries oxygen, and we have normal hemoglobin here. Let's look at what happens with sickle cell. The only thing different in the amino acid chain is the amino acid in the number 6 spot. So in normal hemoglobin, that's glutamine. That glutamine has been substituted for valine, and that's the only difference. And so now, when these guys do the alpha helix and bend and so on to give it its tertiary structure, it has a different shape than what it's supposed to be. And so that both of the beta subunits are made this way, so then we have both beta subunits. These guys are made okay, but unfortunately half the molecule is abnormal now and has a different shape. And so now what they do is they actually bond together, they crystallize, and that affects its ability. You can see here it crystallizes into a fiber and affects its ability to carry oxygen. And so all of those, back to that picture here, that's what's happening to the hemoglobin in this red blood cell, and so it affects the shape of the red blood cell as well, and greatly reduces the oxygen.
Where do those alpha particles come from?
So that's a good question. So we're going to talk about DNA today. There are different genes in DNA. So this is a DNA molecule. Let's say this is gene A here, and then over here is gene B. DNA has lots of different genes on them. So there is a gene that codes for making the beta subunit, and there's another gene that codes for making the alpha subunit. So therefore, there's another whole gene that will make another whole polypeptide chain. Does that make sense?
Yeah.
All right, so that just illustrates the importance. And so when we talk about the shape here, it says exposed hydrophobic region and so on. When we talk about the shape going back to the interactions between the R groups, this is, let's say here, you have an R group and an R group that's supposed to interact. And here we substituted one amino acid for another. So if we substituted and got rid of this amino acid in this spot and replaced it with another amino acid with a different R group, that R group may not bond with this R group, which means then that you won't have that curve, which means then that it'll look like this, all right? And so you wouldn't have that curve. And so that's what's happening here. When you substitute one amino acid for another, if it's an amino acid that's important in stabilizing one of those curves, then you've got an issue. All right, so so moving on here. So proteins are real finicky in terms of their environment. So in addition to primary structure, physical and chemical conditions affect the shape of the protein. So if we alternate pH, salt, temperature, or other environmental factors that can cause a protein to unravel. And so that means, when I say unravel, that means losing its shape. So the loss of that shape is called denaturation, and we call that a denatured protein. And it doesn't work because it doesn't have its right shape. So if you look at, I don't know what I did to this slide and why it does that randomly. I must have done something to that particular slide. So here's your normal protein. It has its right shape and so on. So you have like this curve stabilized by the R groups of these two, amino acids and so on. So every time you see a curve, there's those R group interactions. And so that's your normal protein with the right shape and therefore can do its right job. We said that salt and pH can change those interactions. So salt, like table salt, dissociates into sodium and chloride ions. Or if we change the pH, we're changing the hydrogen ion concentrations, either more or less hydrogen ions. These ions, if they're in the solution of the protein, these ions can actually interact with the R groups as well. So these ions can actually disrupt the R groups bonding and therefore cause it to unravel and lose its shape. So, so that's why our body, we work really hard. We have those buffers that we talked about to minimize resistance, the change in pH, to keep it constant, in part because proteins are really, really insensitive to that. The third thing that we just wrote in one of the bullets is temperature. So temperature, let's talk about high temperature. High temperature can also cause proteins to denature. Why is that? Does anybody have an idea? High temperature specifically, it doesn't apply to low temperature. Think about what's different between high and low temperature and molecules and atoms. Grayson, you think you can? Yeah, the atoms move faster. Remember, this is a chain of amino acids made out of a bunch of atoms, so the kinetic energy increases with increased temperature. And guess what? If it increases so much, these R groups, the interaction can actually break apart, all right? And so therefore the proteins are denatured. That's one of the big things with getting a high temperature. If your body temperature gets too high, you're trying to bring it down. It can be life-threatening if you get it really high, and that's the reason why. It's because you're so hot. All of these enzymes, enzymes are proteins. Enzymes speed up chemical reactions so that they occur at a rate to keep you alive. And so if all of those enzymes, if the temperature gets too high and they start denaturing, you're in trouble because those chemical reactions that they speed up aren't going to happen, and that can be life-threatening. And so high temperature is a big deal. Low temperature doesn't denature the protein because low temperature just slows the molecules down, but it's not going to denature it. But it will slow the reactions down, because if the enzyme is moving slower and it's supposed to like cause a reaction with molecule A over here, the likelihood that the enzyme is going to collide with molecule A goes down when the temperature goes down, but it doesn't denature the protein. All right, and so in studying proteins, like we know a lot about a lot of our proteins now. We've studied them for years. It's hard to predict if we know the chain of amino acids. It's hard to predict how they're going to coil and fold and give their ultimate shape. And so they think that proteins go through several states to get to that stable shape that is going to allow it to do its function. And they're still studying this for a lot of proteins. And they discovered another protein called a chaperonin. These guys assist with folding of other proteins. So it's a protein that helps make and fold other proteins. And so, that's what it looks like here. I want to show you this. So what they found is this is the chaperonin right here, this little guy here. And here's the polypeptide chain. It's just the chain of amino acids. It's not folded in the right shape yet. This protein has a hollow cylinder that the polypeptide goes in. And what that does, the chaperonin, the cap goes on, and they discovered that it helps for the folding, the proper folding of the polypeptide chain, and then the corrective protein leaves, and then it can go and do its job. If it's a finished protein here, if it's at the tertiary structure, or if it's something like hemoglobin, it will have to bond with the 4 other subunits. And so that's something that they found that helps to get it in that shape. The next couple things, I'm going to have you guys don't need to know this, so you can cross off the X-ray crystallography and the NMR, all right? You don't need to know that, all right? We're going to go right on to nucleic acids. All right, DNA is one nucleic acid. What's the other nucleic acid? RNA, right? And so we're going to talk a little bit about the role of DNA and RNA, and then the construction of them, and the monomers, and so on, and how we put them together. So genes are made up of DNA, and as I just erased that. I just erased that. But remember, I just drew here that DNA, sections of the DNA are called a gene, and each gene makes a different protein. So gene 1 codes for making protein 1, and gene 2 codes for making protein 2. All right, and so on and so forth. So we have thousands and thousands and thousands of genes that make all of our different proteins, and those proteins give us all of our traits, all right? From how tall we are, to our digestive enzymes, to the color of our hair, and so on. All due to what proteins we make. So, as we said, there are 2 main classes of nucleic acids, DNA and RNA, deoxyribonucleic acid and ribonucleic acid. And DNA, we'll talk a little bit more about this, provides direction for its own replication. So we can replicate our DNA. It directs the synthesis or making of mRNA. Hopefully you remember that rings a bell, and through mRNA controls protein synthesis, or the making of proteins. And proteins are made in ribosomes in the cell. So that's what the next picture illustrates, all of these bullets here. So doing a quick little review of this. So here you have your cell. This picture is your cell. Here's your cytoplasm out here. This is the nucleus. The nucleus contains your genetic material, which is DNA, which is a double-stranded double helix here. One thing about the nucleus has pores that allow things in and out of the nucleus. So then what happens is DNA contains instructions to make, so there's a gene, let's say right here, and this carries instructions to make a protein. I'll write, I guess I should write polypeptide, that's more correct. Because sometimes proteins are more than one polypeptide. All right, and so this is where the instructions are. So this is like the recipe. The place that the proteins are made is out here in the ribosome. So the polypeptide is made here. So we have a problem because our instructions are in the nucleus, and the place that we need to make the polypeptide is out here in the ribosome, in the cytoplasm. And the thing is, DNA does not leave the nucleus. DNA is stuck in the nucleus. It's too big of a molecule. It cannot get through the pores. In comes the role of mRNA. So mRNA, remember, stands for messenger. So the instructions are copied onto the mRNA. And the instructions here are copied. mRNA can fit through the pores of the nucleus and leave. So that's why it's called messenger. It's taking the message, the instructions from the DNA, and the mRNA goes to the ribosome, where it's going to be used. Those instructions are going to be quote unquote read, and the instructions are going to be used to put what amino acid, so it tells it what amino acid hooked to what amino acid and so on to get your finished polypeptide there. And so, so that's how DNA and a type of RNA called mRNA kind of go together. Do you guys remember this a little bit from bio? Yeah. All right. Yeah? No? All right. So that's a relationship. Don't worry. There's a whole chapter about this. All right. So we're going to learn more about this. All right. Later on. That was just a little overview to just kind of give you an idea of the role of DNA and RNA in the cell. So let's look at the structure of DNA and RNA. They're called nucleic acids. They are polymers. That means they're chains of long, smaller things. And so we call them polynucleotides. Why? Because the monomer is a nucleotide. So therefore, there are many nucleotides hooked together. Now, what is a nucleotide? It has a nitrogenous base, a Pento sugar, and a phosphate group. Nitrogenous base, called that because it contains nitrogen, right? And it actually has the amino groups, which also gives it the base properties. A Pento sugar, pentose, what is that telling us about it? 5 what? 5 carbon. It's a 5 carbon sugar. And then the phosphate group is the P with the 4 O's, right? That was one of our functional groups. And a nucleotide without the phosphate group is called a nucleoside. So let's look at the picture here that shows that. So here's the construction of the nucleotide first. So here's your phosphate group. Here's your Pento sugar. Here, we look here, this is the first 1 carbon, 2 carbons, 3, 4, and the 5th carbon is not part of the ring. These numbers, 3 and 5, are important, the number of the carbons here. I'll talk about that in just a second. And here's your nitrogenous base. And the bases for a nucleotide, it's the nitrogenous bases that can change from one nucleotide to another. We're going to see that there are multiple bases there. So we hook these together. So here's your phosphate, sugar, and base. So this is one nucleotide. This is the bond between the nucleotides. So it's between the sugar of one nucleotide and the phosphate group of the next. So this molecule in this picture, how many nucleotides long is it? Get an answer in your head. How many nucleotides long is this? Somebody want to tell me?
5.
It is not 5. It's not 5. It'd be 5 if I included this one here, but that one's already included in here. This is 4. All All right, so here the phosphate sugar base, here's the bond to the next one. So that's 1, 2, phosphate sugar base, that's the third one bonded here, phosphate sugar base, that's the fourth one. And so this is a polynucleotide. So what do you suppose, where I highlighted these lines, what do you suppose happens there to hook them together? OH and H leave, and that we call that dehydration, right? So we hook them together just like all the other molecules that we've talked about. Let me just point out this, they call this the 5' end of the chain and 3' end. Why that is, if we look back on the carbons, this was 1, 2, third carbon, fourth, fifth, not part of the ring. So they call this the 5' end of the chain because the fifth carbon of the sugar is kind of hanging off the end. And this is the 3' because it's the 1, 2, third carbon kind of hanging off the end. I'm just going to introduce that now because that comes later on when we talk about DNA replication and protein synthesis. That is really important, that 5 and 3'. So these monomers, the nucleotide monomers, made up of the nucleoside and the phosphate group. And remember, the nucleoside is the base and the sugar. Let's talk about the different bases. I wrote on there that the base can change from nucleotide to nucleotide because there are different kinds of bases. So we have 2 categories of bases, pyrimidines and purines. Pyrimidines have a single 6-member ring. Purines have 1 ring fused to another ring. So this one has 2 fused rings. So that's what we're looking at, 1 ring versus 2. And then another thing that differs between the nucleotides that make up DNA and RNA is the sugar, and that's why that's where the name difference comes from. Deoxyribose, ribonucleic acid has deoxyribose as the sugar, and RNA has ribose. So if we look here, that's what we see here. So these 2 boxes, nitrogenous bases and the Pento Sugars. So we're looking at the different types of each one of these. So notice that there are 5 different bases. So these guys are 1 ring. These guys are the 2 fused rings. So 1, 2 sharing a side. And when you see this as a ring, some people think that, oh, it's a sugar, and because it's ring shaped. Sugars don't have what? Nitrogen. So these guys are the nitrogenous bases. And so, so you probably remember, do you remember A, T, C's, and G's from DNA? So they shorthand cytosine with the C and thymine with the T. Notice that thymine, it's important, this is in DNA. This is the base in DNA, meaning that it's not in RNA. And then uracil, U, is in RNA, but not in DNA. That's a big difference between DNA and RNA, is the base difference. Purines with the 2 rings, you have adenine and guanine. And so, so then for your Pento Sugars, we have our 5 carbons. What's different between deoxyribose and ribose? What's one difference between the structure here? What's the difference? See the Derek?
Isn't it the U? Like there's the U in RNA?
Yeah, there is a U in RNA. So, but I'm talking about specifically this deoxyribose sugar and this ribose sugar. Like looking at specifically these 2. This is found in RNA. This is found in DNA. Go ahead.
There's another oxygen in RNA on the lower right. That's an OH.
Yeah, there's another. See how this oxygen here is missing right here? That's the only difference. So deoxyribose is like ribose without an oxygen. That's a deoxy is missing an oxygen. Like when you decapitate something, you take the cap off, or like, and they use it for us like that. All right, the same kind of idea. The oxygen is missing in deoxyribose. So what happens is when we look at like DNA has the phosphate group for the nucleotide, the pentose sugar deoxyribose. I'm just going to put D here. And then you have the base coming out here, and the base for DNA could be C, T, A, or G. So it could have an A, a C, a G, or a T. And so therefore for DNA there's 4 different nucleotides, all right, depending upon what base is attached to it. Got that? For RNA, is also made up of nucleotides, phosphate, and then instead of deoxyribose, the sugar is ribose. And then you also have a base. That base could be one of 4 different bases. You have A, or C, or G, or U. And so there are 4 different nucleotides. And so we hook them all together, and that's how we make either DNA or RNA. All right. So we hook those, well, we just took these monomers together, building a polynucleotide. And this just shows in written format how they're hooked together. So in that previous picture, I drew the bonds. It's the bond between nucleotides are between the OH group of 1 carbon on the nucleotide and the phosphate group of the on the next nucleotide. So I illustrated that with a line. And then what happens is you get a backbone of phosphate sugar units. So when we look at phosphate sugar base A, if this is DNA, this sugar is going to bond with the phosphate group of the next. So here's your sugar, again, deoxy, and then here you have a T, and so on and so forth. And so you keep on hooking the nucleotides together. This is called, right here, the backbone of the molecule. That's why it says the backbone is phosphate sugar bases, and what sticks out are the A's and the T's, and the C's and the G's, and so on and so forth. The bases are what sticks out. That's what it means by this, the nitrogenous bases sticking out like appendages, all right? And it's the order of those A's, T's, and G's that determine what protein it's gonna code for and make. And so DNA is a double helix, which means it has 2 polypeptide chains. So you've seen DNA as a double helix, and they have these 2 backbones running in opposite directions from one another. So it's called anti-parallel. And each one of those, the order of those bases, include many genes that can code for many different proteins. And the 2 sides, the 2 polypeptide chains, how they stay together is they're hydrogen bonded to one another, and they always hydrogen bond in the same way. A always goes with T, and G always goes with C. So what would happen is if I were to make another polypeptide chain that would hydrogen bond to this, the base across here would be what? T. And the base across from a T would be an A, and so on. And that's what this picture is showing you. This is the double helix. Did I go too fast?
Yeah.
All right, I'll go back. We're gonna finish it. So this is what we have. Phosphate sugar backbone, bases sticking out, and C's always go with G's, A's always go with T's. So you're familiar with that picture? You remember that picture from before, all right? So the last thing is that the DNA, we're gonna link it to evolution and relatedness between organisms, that the order of bases in the DNA are passed from parents to offspring through egg and sperm cells. And 2 closely related species have more of those bases in common. The order of the bases in common are more similar than organisms that are distantly related. So therefore we use that to tell relatedness among species. Yeah, don't forget your phone. Yeah, okay, I'll take it.